Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please do us a huge favor. Like, subscribe, and share this deep dive with friends and family. That simple act is genuinely how you support the Science Experience and helps us bring cutting-edge research directly to you. So today, we are examining a really critical review. It suggests something that might fundamentally shift how we look at longevity research. When we talk about biological aging, we usually track things like mitochondrial decay, epigenetic clocks, maybe cellular senescence. But this research points out that most current biological age tracking operates under a pretty big assumption that these changes happen in, sterile conditions.
Exactly. And that assumption of sterility, that's precisely the challenge this paper tackles head on.
Yeah.
We're looking at a really comprehensive review of the molecular mechanisms.
Yeah.
It shows how persistent infections, we're talking viral, bacterial, fungal, parasitic, are major, often just completely overlooked, drivers of age acceleration. These chronic agents aren't just, passive passengers alongside aging. They actively hijack the very processes that define it.
Wow. Okay, so we're shifting the question maybe from why are my mitochondria failing to, wait, who is making my mitochondria fail? Let's start by defining what we mean by these persistent actors. What is persistence in this context?
Right. So we're talking about chronic persistence. These are pathogens that basically settle in for the long haul. Every single human sustains infections, but many of these agents are capable of long-term latency. They hide out, often deep in host tissues, in nerves, sometimes even including the central nervous system. And crucially, even when they seem dormant, these latent microbes, they're still active enough. They express proteins that definitely interfere with our host gene expression and, importantly, immune signaling.
And we're not talking about, rare tropical diseases here, are we? This is stuff most of us encounter.
Oh, absolutely. We are talking about the basic human virome, the things pretty much all of us carry around. The core of this persistent human virome is largely made up of herpesviruses. Things like Epstein-Barr virus, human herpesvirus 6, and especially Cytomegalovirus or CMV. These are harbored by over 90% of the human population. They're often silent, yeah, but they seem to be highly influential passengers.
90%, wow.
And it's not just viruses. We also see intracellular bacteria and parasites. Think Toxoplasma gondii, that's harbored by up to maybe 50% of people in some parts of the world.
Okay, and it seems this idea of persistence isn't just for old viruses. It applies to newer things we face, too.
Without a doubt. There are modern studies, very current, that have identified persistent SARS-CoV-2 RNA, or sometimes the spike protein itself, in dozens of different human tissue types. And this is weeks or even months after the initial acute infection was supposedly cleared, even people who had no symptoms to begin with. So yeah, it really reinforces this idea that persistence is a fundamental microbial strategy, and we absolutely have to integrate that into how we view age-related health and disease.
Okay, so if these microbes are sort of hidden away in our organs or nerves, what are the actual blueprints they're using to speed up aging? What are the key molecular mechanisms connecting, say, a latent herpes virus tucked away somewhere to an older biological state?
Well, the first and probably the most recognized pathway is inflammaging. That chronic low-grade inflammation that's often associated with aging. It sounds kind of sterile, right?
Right, it just sort of happens.
Yeah, but what we now understand is that it's heavily fueled by these persistent pathogen proteins. These proteins basically continuously irritate the immune system across different body sites. They drive up pro-inflammatory molecules like cytokines.
Okay, can you give us a concrete example? How does a viral protein trigger the specific inflammation?
Sure. Let's take the cytokine IL-11, interleukin-11. It's known to be upregulated specifically in response to various viral pathogens. And researchers found that when they inhibited IL-11 signaling in mice, it extended their median lifespan by over 22%.
22%, just from blocking one inflammatory signal linked to viruses.
Exactly. That single data point really suggests that controlling just one aspect of this pathogen-driven inflammation could be a really powerful lever for longevity.
That brings up a huge point then about, immune bandwidth. If our immune system is constantly dealing with this low-grade pathogen-driven fire, that must drain its resources, right?
It absolutely does. Think about it. Chronic infections mean sustained antigen exposure. The immune system literally never gets a break, and this leads directly to something called T-cell exhaustion. Basically, the T-cells are key immune fighters. They lose their functional ability. They can't proliferate properly anymore.
So they just get worn out fighting these persistent threats.
Worn out is a good way to put it. And get this. With chronic CMV infection alone, which again, most people have, approximately 10%, one-tenth of a person's entire T-cell population can end up being directed against just that single virus.
Wait, 10% of all our T cells just fighting latent CMV?
Precisely. So think what that does. It depletes the pool of naive T cells you need for new threats. It forces the immune system to divert massive resources constantly. It accelerates immunosenescence, the aging of the immune system itself. And your capacity to fight off new things, like maybe influenza or even cancer, is likely severely diminished.
Okay, that's a huge impact. Now let's shift gears slightly to how these pathogens mess with our internal machinery. We often talk about mitochondrial dysfunction as a hallmark of aging, but this research suggests it's less dysfunction and more theft, like the mitochondria are being actively stolen.
That's actually a great way to put it. Viruses and these intracellular pathogens, they are obligate parasites. They have to hijack the host cell's mitochondria. They need access to the fatty acids, the nucleic acid building blocks, all the substrates required for their own replication.
Yeah.
And they achieve this by fundamentally reprogramming the cell's energy production.
Okay, how do they pull off that metabolic switch?
They induce what's called a pathological Warburg metabolic shift. So normally our cells prefer oxidative phosphorylation, RxPHOS. It's very efficient energy production using oxygen. But the pathogen forces the cell towards preferential aerobic glycolysis. It's much faster, sure, but way less energy efficient overall.
Why would they do that? Sacrifice efficiency?
Because the pathogen doesn't care about the host's long-term energy efficiency. It needs the raw materials, the glycolytic intermediates that this rapid pathway produces. It needs them now for quick replication. Viruses like Cytomegalovirus, parasites like Toxoplasma gondii, they employ this exact strategy, hijack metabolism for building blocks.
Right, and if they're hijacking the energy system, it makes sense they'd also go after the master regulators. The pathway is designed to keep the cell healthy and long-lived.
Oh, absolutely. It's a key part of their strategy. Viruses have actually evolved very specific proteins to co-opt key longevity pathways. A major target is the mTOR signaling pathway. mTOR controls so much cell growth, division, mitochondrial function, autophagy. It's central.
So they hack mTOR.
They do. There's a detailed study that found proteins from 122 different viruses, including really common ones like influenza A, all converged to interact with 69 specific human proteins already linked to aging. And the highest rates of these interactions, they were linked specifically to proteins within that mTOR signaling network. It confirms viruses are practically hardwired to disrupt our longevity circuits.
Okay, this next connection is, I think, really critical for listeners, especially thinking about diseases of aging. You're linking this chronic microbial activity directly to neurodegenerative diseases like Alzheimer's and Parkinson's. The data suggests infections maybe decades earlier could set the stage.
The epidemiological evidence is becoming quite compelling, yes. Several studies show that individuals who had hospital-treated infections serious enough to need hospital care in early or midlife, they had a significantly increased risk of developing Alzheimer's and Parkinson's disease maybe 5 to 10 years later down the line. And having multiple infections before the age of 40 seemed to confer the greatest risk, which really underscores this idea of a cumulative burden over time.
Wow. And this leads to what I found maybe the most thought-provoking idea in the review. Our whole understanding of the pathology itself might be inverted. Like amyloid-beta plaque, the hallmark of Alzheimer's. Maybe it isn't just garbage waste buildup. Maybe it's actually a failed defense mechanism.
That's the key shift in perspective. Exactly. Amyloid-beta is increasingly understood, based on functional studies, to act as an innate immune system antimicrobial peptide, like an antibiotic produced by our own brain. The thinking is it forms rapidly as a protective response. It seeds around invading pathogens, viruses, bacteria, even fungi to physically entrap them. We see this documented quite clearly now with pathogens like herpes simplex virus 1, that's the cold sore virus, and also HHV6. The plaque forms around the microbe. It starts as a shield.
That completely shifts the goalposts, doesn't it? If the plaque is a defense mechanism, then maybe we've spent decades trying to clear away the immune response rather than tackling the chronic threat that triggered it in the first place.
It certainly reframes the therapeutic challenge.
Yeah.
And the specific finding linking CMV, cytomegalovirus, is powerful here too. Chronic CMV infection has been directly linked in some studies to accelerated Alzheimer's-related pathologies. And crucially, CMV protein itself has been found in the gut tissue, along the vagus nerve, and actually in the brain tissue of Alzheimer's patients.
The vagus nerve. So that's a potential highway from the gut to the brain.
It offers a plausible pathway, yes, for how a persistent gut or systemic infection could potentially seed the brain over a long period.
Okay, and for Parkinson's disease, we see a similar story, but with a different protein, alpha-synuclein.
Yes, there's a parallel concept. Alpha-synuclein, the protein that aggregates in Parkinson's, also shows antibacterial activity. It seemed to play a defensive role, too. And very current research, again involving SARS-CoV-2, showed that the spike protein specifically can induce neuroinflammation and trigger the accumulation of alpha-synuclein in brain regions that are relevant to Parkinson's development. So it strongly supports this idea. Persistent infection isn't just correlated, it seems to directly fuel the pathologies underlying these devastating neurodegenerative diseases.
Okay, so if chronic infection is looking like a core driver of aging, maybe even the driver in some contexts,
Yeah.
what does this mean for health span interventions people are already exploring? Does it suggest some popular longevity compounds might be working, at least partly because they're acting as sort of hidden anti-infectives?
That's exactly the implication. And it makes sense of some existing data. Let's look at targeted therapies first. The data for standard antivirals, particularly antiherpetics, is incredibly strong in some studies. Research on patients with documented chronic HSV infections, herpes simplex, who were put on antiherpetic medications, showed really an astonishing reduction in subsequent dementia risk. Almost 91% reduction in one large study, especially if the treatment course lasted 30 days or more.
90% reduction in dementia risk just from treating herpes. That's incredibly specific evidence for the infection model.
It's very compelling, yes. Highly specific. But then let's look at the broader compounds we hear about in longevity circles.
Like rapamycin or metformin.
Exactly. Take low-dose rapamycin. It's primarily known as an mTOR inhibitor, regulating cell growth and autophagy. But clinically, it's also been shown to improve oral health. And remember, periodontitis, gum disease, is fundamentally a pathogen-driven inflammatory disease. Plus, by inhibiting MTO, rapamycin also seems to help reduce markers of that T-cell exhaustion we talked about. It might allow the immune system to regain some bandwidth to better handle persistent infections.
Interesting. Okay. And metformin, often thought of for blood sugar, but you're saying it has antiviral properties.
Correct. Metformin activates a key cellular energy sensor called AMPK. This activation essentially depletes the cellular energy reserves, the ATP, that many viruses, including HHV6A, absolutely require for replication. So, in effect, metformin might be starving the virus of the energy it needs to copy itself. It acts as a broad-spectrum antiviral in that sense.
And NAD plus obo, that's another big one in longevity.
NAD plus replenishment, yeah. This seems to work partly by supporting the overall immune response. It also helps activate antiviral interferon-stimulating genes directly. Plus, many pathogens actively deplete host NAD plus levels as a survival tactic. So boosting NAD plus could directly counteract that viral strategy.
Okay, so we have the why, the molecular mechanisms linking an infection to aging. And we have hints of the what works interventions that might control these hidden infections, maybe unintentionally. But when we try to apply this clinically, we immediately hit a massive wall, don't we? And that's the diagnostic challenge. How do we even know who has which active persistent infection?
That is the absolute critical bottleneck right now. It's the biggest hurdle. Remember, these persistent pathogens hide deep in tissues in nerves. They are generally not freely circulating in the blood where we usually look. They are tissue resident. So standard blood tests like measuring IgG or IgM antibodies, they're often woefully inadequate for detecting active tissue-level persistence.
Right. Why are those traditional antibody tests failing us here, especially given, as you said, almost everyone tests positive for IgG to something like CMV anyway? What's missing?
What's missing is the understanding that these pathogens are masters of immune manipulation. It's how they survive long term. They actively dysregulate and suppress the host immune response. So paradoxically, a patient might have a very intense, active, persistent infection-driving pathology in their tissues, but they might fail to mount a robust IgM antibody response in the blood, or their IgG profile just tells us, yeah, they were exposed years ago, not this thing is actively replicating in the nervous system right now.
So the blood test doesn't reflect the tissue reality.
Precisely. We desperately need reliable proxies for active tissue residence. We need to know what's happening locally, not just systemically.
Okay, so clearly we need to move beyond the standard immunology lab tests for this. What are the cutting-edge research tools that might actually bridge this diagnostic gap, make these hidden infections visible to a doctor?
Researchers are developing some incredibly sophisticated tools. It's an active area. One approach is using ultrasensitive protein assays. Technologies like Simoa single-molecule array can detect minuscule amounts, like femtogram levels, of specific pathogen proteins that might be leaking from tissue reservoirs into the blood. This lets us spot things like tiny amounts of circulating SARS-CoV-2 spike protein long after infection, something a standard test would completely miss.
That level of sensitivity sounds key. What else is emerging?
Another powerful tool is cfRNA metagenomics. That's cell-free RNA. This allows for unbiased detection. Instead of looking for one specific virus you suspect, you sequence all the microbial RNA fragments floating in, say, plasma that came from tissues. This tells you which microbes are actually transcriptionally active right now, anywhere in the body.
Like a snapshot of all active infections.
Kind of, yeah. And finally, there's deeper immune repertoire profiling. Not just IgG levels, but tracking very specific antibody responses. For example, researchers are identifying unique combinations of IgG4 antibody epitopes, tiny parts of the antibody that are specifically raised against CMV. And these specific IgG4 signatures seem to correlate strongly with finding CMV physically present in Alzheimer's brain tissue.
Ah, so if those blood-based signatures hold up, that could be a game changer for accessibility.
Exactly. If we can validate those kinds of blood-based signatures for tissue-resident activity, we might finally solve the diagnostic accessibility problem.
This has been a truly fascinating deep dive into what feels like a hidden landscape of aging drivers. I think the core conclusion we really have to take away is that incorporating persistent infection into our fundamental models of aging is absolutely essential. We can't ignore it if we want to optimize therapeutic strategies. The focus really needs to shift, maybe from just managing the downstream symptoms of aging to actively identifying and controlling the infectious inputs that seem to be accelerating the whole process.
And perhaps this raises an important, maybe provocative question for you, the listener, to consider. If chronic tissue-resident infection really is a fundamental lifelong driver of the aging process for many, perhaps most people, what incredible new era of truly personalized, pathogen-specific longevity medicine might emerge once we finally develop the tools to accurately diagnose and manage these active microbes across a person's entire lifespan?
That is a powerful challenge and maybe a hopeful one for the future of health. Thank you so much for joining us for this deep dive into the intersection of infection and aging. And please don't forget to like, subscribe, and share this information with anyone you know who's curious about health, aging, and longevity. That support really does help the Sans Science experience. We'll see you next time.